So let me introduce you to American Gene Technologies, a company in Rockville, Maryland, and with a focus on lentiviral vectors for important diseases in man. And today I'm going to discuss our current program at AGT, which consists of three major focus areas. HIV at the top, PKU next, and liver cancer are an immuno-oncology program shown at the bottom. And I'm going to focus mainly today on the HIV asset. As you can see, that's scheduled to have IND submission and enter the clinic in Q1 of next year. We're quite excited about this program. We think we have a real shot at achieving a functional cure for HIV. And we think it's an innovative program that brings something new to that space. If I have time at the end, I'll briefly mention what we've been doing in PKU and the immuno-oncology work, because I think those are also quite interesting programs. The functional cure for HIV remains an unmet need in medicine, and suffering continues in these individuals long after they go on stable antiretroviral therapy with viral suppression. We need a way to fully reconstitute the immune system in these individuals to reduce the rates of comorbidities and perhaps allow an exit from lifelong dependence on chemotherapy. And our program is designed in this way to be an immunotherapeutic combined with a lentiviral vector strategy in order to deliver something of great value to these individuals. There are really three components to the product that we're making. We know that we need to enrich massively for HIV-specific CD4 T cells. The loss of this T cell subset is the true hallmark of HIV pathogenesis. During acute infection, these cells are lost at a preferential rate much more quickly than the bulk rate of CD4 T cell depletion. And if one reconstitutes these cells through long-term therapy or tries to boost them with therapeutic vaccines, the reemergence of virus once therapy is removed will again deplete these cells at a massively accelerated rate. This is the key defect in HIV, in my mind, and I believe much of the literature has focused on this over the years, but nobody's really developed a strategy for going after it. We think the enrichment for HIV-specific CD4 cells in a cell product is the key issue that helps us close the gap between functional cure and the programs that have come before us, which made progress but didn't quite make it to the line. Inimical to this program is we require a very potent lentiviral vector, because if we enrich for HIV-specific CD4 cells and return them back into the body, they will simply be attacked by HIV and depleted again. So once we enrich them ex vivo, we have to then protect them from a future depletion event by modifying them with this lentivirus vector. And I'll describe the vector and its potency in a minute. And then we infuse these autologous cells back into the original donor in an effort to build a central memory T cell help capacity against HIV that is in the numerical range that we expect for normal central memory able to control other types of persistent viruses. The process is, it's a complex cell process, as you can imagine. We're working on the cell process development with Miltenyi Biotech in Sunnyvale, California. And we have the option at the beginning of this process to incorporate a therapeutic vaccination to pre-boost those HIV-specific responses. And that vaccine has been obtained in a clinical collaboration with GeoVax Incorporated in Atlanta, Georgia. The process is to leukapheresis these individuals, put them on the Miltenyi Prodigy machine for about a 15-day process that involves stimulation with peptides that represent viral proteins, and then transduction into the cell population. There's a few other earlier steps that are required to increase the yield of the specific cells that we want. And most importantly, these cells are vanishingly low levels in patients, despite even years of antiretroviral therapy. And the vanishing low levels shown this is a fairly typical individual here, about 0.055% of starting frequency. This is terrible in terms of viral immunity, but it's common for HIV patients. But in this process, we can amplify that up to about 7.5% of the CD4 cells in the dose. And when we return that dose, we should engraft up to a level of about 0.1 to 0.3, maybe 0.5% of total body CD4 cells will now be memory against HIV and protected from subsequent depletion because of the introduction of our lentivirus vector. So this is a passive immunization strategy based on CD4 T cells, but with the important trick that you have to introduce a protection against HIV, or the progress you make in enriching and infusing will be lost quite quickly. Here's another example. This is a person that starts with an extremely low percentage, 0.027%. It's right at the bottom of what you can actually measure in these type of assays. And in a 13-day process, we can get this up to 6.5% of total CD4 cells. And we think that enrichment compares favorably for what's going on in enriched T cell products against other infectious diseases and against tumors. So the potency has been tested in a number of ways, and our lentivirus vector has 3 elements on it. We're downregulating CCR5, and we use an inhibitory RNA to do this because we think that's more efficient than other types of knockdowns. So the inhibitory RNA will reduce CCR5 expression to undetectable in 100% of the transduced cells. However, we want to protect also against CXCR4 tropic viruses and or viruses that bypass the CCR5 block. So we've included 2 inhibitory RNAs against HIV, and we calculate that they will attack about 70 to 80% of total clade B sequences. You can see that the potency on the left is showing you what happens when you use a lentivirus vector, our lentivirus vector for protection, followed by challenge with a CCR5 tropic HIV. And you can see that you reduce the number of infected cells from about 54% here down to less than 1%, so that's a very substantial protection. And on the right, it shows that you get almost the same degree of protection when you do this against the CXCR4 tropic virus. Now, very importantly, this would not be a drug, right? A drug would have to give you 5 logs of reduction of HIV, something like that, in order to be effective. Here, we're just trying to protect a high enough proportion of these cells to allow them to respond to antigen if and when HIV reemerges, and to proliferate and function normally. And so we don't have to protect 100%. We just have to reserve a good, strong population capable of doing their job when the time is called. I also want to remind everybody of a really important difference between CD4 T cell responses and CD8 T cell responses. We focus a lot on the CTL, and we focus a lot on HIV variation escaping the CTL. That's because CTL recognition of peptide epitopes is stringent. Single amino acids can eliminate the recognition, and it's very restricted to individual MHC haplotypes. On the CD4 T cell side, recognition of peptide epitopes is much more degenerate, and it tolerates changes within the sequence, and individual peptides can be dominant across a wide range of human HLA haplotypes. So working on the CD4 T cell side, we don't need to have peptides or anything else matched to the autologous virus in that individual. We simply need to get a good, strong, positive stimulation, and we're achieving the types of stimulations that I showed you before routinely in a very high percentage of patients that we've surveyed so far. To give you an insight into the timeline on this program, we've just released the cGMP vector that came from Lentigen in Gaithersburg, Maryland. The IND submission is well on track for January, February time of 2018. We've identified 2 trial sites. One will be in Baltimore and one will be in Washington, D.C. And we've already launched a clinicaltrials.gov activity at one of the trial sites to collect material, clinical material, for the final process development steps. We think we'll read interim safety by Q2 of 2018 and start to get interim data on the ability of this therapy to reduce markers of infection by heading towards the middle of 2018. And the markers we're focusing on mainly are the size of the viral reservoir, the functionality of the CD4 and CD8 T cell populations against HIV, and the immune exhaustion markers that are common, especially in CD8 cells. And the mechanism of action that we're really looking forward to here is that with this improved T cell help, we will be able to drive the differentiation and activation of new CTL clones at a rate sufficient to counteract the viral variation rate. There's nothing special about the mutation rate in HIV. It's exactly like all other RNA viruses, and most of them we deal with quite well. The key in HIV is the disabling of the T cell help, causes a failure of the CTL response, which results in escape and lifelong infection. So we believe this is a very potent way to go after the key step in HIV pathogenesis, which we all knew about for a long time and stumbled on as we were going after other more exotic therapies. So let me just take a minute or so and talk about the other two programs just so you get an introduction into this. We've been focusing on a lentivirus vectored phenylalanine hydroxylase gene for PKU therapy. This was a tour de force of engineering, because if you know about this area of work, PAH is a notoriously difficult thing to express. The human gene is really difficult to express. So after about 1 year and about 100 vectors into this program, we got a very strong series of expressing PAH vectors, and that express both in cell lines and express at very high levels with strong enzymatic activity in primary human hepatocytes. And virtually every part of this locus had to be re-engineered. I have really to this day no idea really how this thing is regulated in vivo. It's really a mess. The very first generation vectors, which were about 1% expression level of these, had already shown small potency in neonatal mice for the PKU model, correcting the growth defect and giving us some substantial reduction in blood phenylalanine levels. And as we go forward, we hope that the more potent vectors will have an equally potent effect on mice, and we're expecting that data to come in the next 2 or 3 weeks. So PKU is an active program for us, and we like the idea of lentivirus vectors for permanent modification of the liver, with the possibility to eventually go into children with a single dose of vector that will then grow and scale as the liver grows in children. Just to quickly introduce our immuno-oncology program, I think this is something quite innovative. And what we're doing is we've developed lentivirus vectors that alter the metabolism of tumor cells. And by introducing these metabolic defects, these tumor cells then become highly stimulatory for the circulating dominant population of gamma delta T cells in blood. And that stimulatory activity will cause those cells to proliferate and become potently cytotoxic. Now, if you know anything about that system, gamma delta cells use a T cell receptor for specificity of stimulation, but they use NK-like receptors for tumor recognition and killing. So if we can get vector into a small fraction of tumor cells in a solid tumor, we should be able to stimulate a response that's potent enough to kill the rest of the cells, because you don't need the genetic modification to kill. You only need it to stimulate the gamma deltas. So in these very early mouse experiments that we've been doing, we grow tumors to a pretty large size containing the lentivirus vector. Then we inject human PBMC, and as you can see, get complete eradication of substantial tumors. And right now we're running about 80+% survival, about half of those being complete remissions in the mouse studies. So we're very excited about this approach because it has a couple of unique features. We only target the tumor. We don't modify the immune system. Gamma delta cells have never been associated with autoimmune disease or any immunotoxicity known, and we believe that once the tumor is eradicated, the drive of the gamma deltas is eradicated, and the system will go back to normal. This should work across a broad range of tumors because gamma deltas are not very specific and they will kill many different tumor types. So this is very actively in development in the company also. And as I said before, we're very excited about crossing the clinical threshold early next year with our HIV program, and we see strong progress in these other areas as well. So thank you very much. Oh, I forgot to show you our team. On the left side is Jeff Galvin, who's here in the audience, so say hi to Jeff. That's our CEO. So.